Our speaker now is Dr. Akhil Ranjan Baruah. He's assistant professor of our department. He did his master's from our university, and then after getting a fellowship from named as the Japan Government Fellowship next for his PhD degree, he went to Hokkaido University in Japan. He spent there for almost 5 years, right? More than 5 years doing his PhD work, and he has done excellent work. The work was published in very new phytopathologist, right? Yeah, some of the new very high impact factor journals. Then he came back and joined our university as assistant professor. And since then, he's working on plant molecular genetics, genomics, as well as transcriptomics. He has several projects from DBT. And he is basically working on rice. Rice is a major crop he is working on, and mostly abiotic stress management in rice. And he has been engaged in elucidating the genetics of cold temperature adaptation in Boro rice and broad adaptation in upland rice of Assam. So I welcome Akhil Baruah again. You okay?
Yeah, thank you.
Over to you.
Thank you so much. Thank you so much, Dr. Sumita Acharya. So she's senior to me. I think you have been a very nice time here. Probably some of belong to our university and some of you are from different universities across Assam. Anyone from outside Assam? Okay, there are many. And you belong to different universities as well, right? Okay. First of all, I would like to thank the organizers for selecting me or recognizing me as a resource person for this kind of workshop. So I'll try to justify because most of you are all students. So I'll try to justify some of the ideas that we have in our mind. Okay, my topic of discussion, as you can see in the topic, that we know that what Mendel has contributed to the field of genetics. We all know who is Mendel. I think anyone who doesn't know about Gregor Johann Mendel, I think everyone knows it because we read it in our school book as well, right? So his work, we all know it. So now, a paper which has been discussed 130 years before or 130 years back, and now in the year of say 2022, where we have such splendid technologies, technologies from all the sides, all the fields, that same is for our biotechnology as well. There are a lot of new technologies, newer technologies coming up, so we are accustomed to it, or we have to accustom with all those technologies. And now the question is being posed, whether whatever Mendel has given to us is still holding true, or whether Mendel was that much lucky that he got that kind of relations, and he was so, he could have to simplify the stories of, basic genetics or the fundamental genetics in a very simple word. Is that so simple? That is our topic of discussion today. I will try to check. I think you already have gone through probably different field at IRS Davak. Probably in the morning you have the division there, although it was raining, right? You got some games, and there are some varieties probably you can know about. So Dr. Sanjay Shetty probably has some class, right? Did he take some class? Okay. So he might be telling about Swarna Sadhwan. He is also telling about, say, Ranjan Sadhwan, Bahadur Sadhwan. So those where we are interlearning the technologies. So the breeding, conventional breeding was on the way, following the manual. Okay, the green revolution period, it was due to the maturing of SU1. Okay, so it was one thing from DZ to Z. Okay, it came into, it's all follows manual. Manual is called inheritance. That is what we all know that this is the one ratio typically for all the language, right? And then you just go on independent as well. Okay? Then you all know that what Megan has talked about. So now the question comes: why do we need to study this kind of information? Why do we need to, have, or why I have to do this topic? Okay? Why? So when you talk about manual, there were 7 traits, right? There were 7 traits, and most common is being your height of the plant, right? All the time you check a board, you might be, right, like capital VT, small VT, right? Those things you have in your writing, right? Capital VT, small VT, so tall and dwarf, and you make a checkerboard, and you page the ratios. Typically, and then typically. Okay, so for example, so I believe this is the color, I believe the purple and white you put it together. So as you can see here, so it's 3:1 ratio. That is for phenotype. Discrete, very much, discrete. One is purple, another one is white. There is no intermediate. Only 2 traits are involved. So one is the trait is what the color. Trait is one, the color of the flower. But you have only 2 alternatives. One is purple color, and another is also white color. And genotypically, when you say we have 1:2:1, right? And we are getting genotypically 3:1. Okay, by 3 great scientists. Do you know the scientist's name? Anyone? Ranjan published in 1864. It was unnoticed. It went unnoticed, and then it was rediscovered by 3 scientists. Do you know anyone who rediscovered? There were 3 scientists. Yeah, yes. Samar and Vigodi Rice, Samar, and the last one. Correct. Right? So it was the year 1900, right? People get to know that this is what the ratio. Usually, prices segregate. He took Python survival, okay? He is an example. Okay? So people thought that whatever he has found is found to be true. So there are a lot of scientists on that side. They don't agree to it. Because the things that is in the environment or in the outside world is quite complex. Okay? Mental got his ratio, it's fine. He got in his experiment, that is also fine. So for example, height, he got what? 3:1. Okay? 1:2:1 for capital VT, 2 capital V, small v, heterozygous, and then small v. That is your homozygous recessive. Okay. And he got phenotypic 3:1 for tall and dwarf. Out of 4, 3 you obtain as tall and 1 as flat. Fine. Now, height in Python diagram, if we can code it. Can you stand up? Can we stand up? I am standing up. Can you stand up? Oh, video memory. To check each other. Can you differentiate? The Mendel was very lucky. I mean, if you go back, so this is your tall, three tall, one broad. Now, if you can see here, now you can tell. I mean, I can find him as three. I mean then there's like we can go like in a class, right? There's a class. I don't want to discriminate. Someone is short, someone is tall. I sit down. I don't want to discriminate. But the point is, it's very simple. I think it's very simple. 3 is to 1, but we're not getting it. Why should I be like that? Is that the point? So if I'm plotting, okay, it looks like this. Some might be say, in a classroom, might go for that. So one will be having much taller. Maybe numbers are less, but still they are tall. So there is maximum and minimum. Okay, there are ranges. When there are ranges, the traits are multiple. You get some intermediates. Intermediate is what? Say, if I am getting say 4 feet, 4.2, 4.3, 4.6, 5 feet, 5.2, 6 feet. So that means there is no any demarcation. If the range is there, within the range, all the variables are being placed together. So those traits become complex and addressing, utilizing this only manual principle to stick to that. We cannot explain. But again, very important point. We cannot avoid manual. Don't take a perception that, okay, problem on manual. No, this is not the correct example. Manual was right at his own place, in his own experiment, but we cannot relate his experiment to our own purpose all the time. But this, when we want to have, say, we are introducing variety. We are introducing variety to the farmers for a particular trade. So that trade has to undergo more or less 3:1 segregation. We try this complex trade, inner ranges, and from the ranges we need to chop out the base one. So chopping out the base one is not a better option. Okay. Scientists from across the world working, since say 1960s to mix or to get break the yield pattern. So it is not being even possible, but still going on. Just need to break the barrier. So we'll try to check how we can address. So these traits. Okay, one hand, people are struggling for water. In another case, what? People are struggling for water. Like the crop. Okay, the crop is also under stress. So it is very, very. So water stress. One is flooding stress. So these are natural phenomena we cannot avoid. We can count them. We cannot avoid. But that's why the traits that Mendel just talked about don't look simple. So we want to have flood resistance, we want to have drought resistance, but to have both, these traits are not easy to manage. It's not like, another flower, like white and purple, it's not. It is. These traits are very, very complex traits. Why I am referring to complex traits? Because these traits behave almost differently in two different environments. Okay? They are not standard. It may perform here well in, say, Baruah condition. It may not perform well in North Bend, say, Lakhimpur condition. It may work in Assam plain condition, it may not work the plains of West Bengal. It may not work in Kerala. So likewise, there are variations. So how best we can manage? So that is the question. Okay. Now, so how to address this? So to address the complex traits. So basically, the genetics of the complex trait is under polygenetic control. So now question comes, what is polygyny? Okay? Polygenic control. Polygenic control of target. So what is polygen? So metal what he was referring was oligogenes or mesogenes. One gene contributing a major effect. So one gene, one gene. Here there are multiple genes, more than say 2 and more genes, governing a single trait, having cumulative effect. That is very important. Cumulative. The effect of which may vary from greater than 0 to 100%. Meaning what? Say, if you are all genes, so he is contributing say 1% to the trait, so C is contributing say 3% to the trait, C is contributing say 40% of the trait, but effects of all are being contributed. They will see on different parts of the chromosomes. So if it is right, so they are sitting on 12 different chromosomes. 12 different chromosomes. Rice has got 12 haploid chromosomes. So Bison sativum has got 7 haploid chromosomes. Okay. So now these 12 chromosomes, all these will be there. Say if there are 20 genes, all the genes will be there. Okay. So these 20 genes will show different of various languages. Okay. So when the genes are going here and there, multiple genes, so there are probabilities that say there are 4 or 5 measured regions, REGI region, measured region on the chromosome, 4 or 5. 4 or 5 measured regions or locations on the chromosome which may contain genes of interest. Okay. I say there are 20 genes. These 20 genes may contain in 4 or 5 locations of the haploid chromosomes. Say 12 chromosomes, rice has got 12 chromosomes. Out of the 12 chromosomes, say there are 5 chromosomes where the genes are being located. In one location there are 6 genes, another location there are 2 genes, some other location there are say 7 genes like this. Okay. So those regions we usually refer to as quantitative trait process. Okay. So this is what, and for to study this, there is a complex trait, QTL mapping is. So QTL is the locus, it is not the gene. Flanked by certain markers which are linked to the trait of identity. Again, the question comes, what is marker? Question comes, what is marker? I told that QTL is a locus, quantitative trait locus. A locus governing a quantitative trait to the very raw definition. But to be very specific, QTL is what? The locus of the chromosome which contain polygenes flanked by tightly named marker. Now question comes, what is marker? So if someone or your friend asks you what is marker, so how do you say? Marker, location of QTL or location of QTL. So what should be a marker?
Particular
landmark. Landmark, yes. Landmark. Good word, landmark. So what is your landmark? So what is your landmark? You are a student. Student of Kaziranga University. So a person sitting at say United States, say he doesn't have Google Map. He doesn't have Google Map, and just wanting to know where are your Google page. Okay, what is it going? If someone wants to know that somehow, like one of your friends sits in Houston, in United States and wants to know more about you, and you want, I mean, that friend sending another friend you this, how he will say his friend to reach you. People,
The country India,
okay,
state Assam,
okay,
district Baruah.
okay.
So then the university, Kazan University, then student.
So he is saying some that's called the landmark. Without having Google Map, now without Google Map we can't even move, right? We can't even imagine, but still we can find out, right? So these are markers at different levels. So for the United States, if you say right, it comes over. Probably he doesn't know, right? But if you give a landmark like India, okay, you understand that. First, I have to go to India. Then, from in India, one English, you say Assam, then you come to Assam. Then from Assam, come to Baruah. Then Baruah to them. So these are kind of a marker. So here also, like in QTL, this is plant biology, okay? Level 3, right? So, sorry. Sorry. So these are the landmarks. So the marker what I talked about. So this marker will just be kind of a sequence. Okay. That sequence is not directly associated with detail, but will be linked, will be tightly associated. What tight association is? So when there is crossing, when you do a crossing, so when it goes to the next generation, it will go together with it. Okay. So there is one marker. So seeing that the marker, we will be knowing that okay, this region of the chromosome is also coming to the next generation of one person. So this is what the QTL mapping. So now this QTL mapping, as I say, here are a lot of effects. Okay, so the size of the effects are varying. Okay, but these are all say contributing. All are contributing towards a particular quantitative trait. So this is your chromosome. So within the chromosome, we have the DNA. So I mean, all the QTLs may be on different locations in the chromosomes contributing towards a particular quantitative trait. And when you go for second procedures, you can detect the region, or you can find out the region. Okay. Now, question comes: how to map the QTL? So, how can you map the QTL? So, there are two ways. Okay. One, we call it as biparental, using biparental mapping population, biparental crossing, or next is your association mapping. Two ways. So biparent is what? Two parents. Bi means two parents. So these two parents, one a standard variety, should be a standard variety, and the other one should have the trait of origin. Say drought cover, say flooding cover. So these right hand side, I mean my right hand, the variety is in my right hand. May not be the standard variety, but this variety has got what? That trait of origin. So this trait I will introduce here in the standard variety. So there is a biparental process, and this cross may be of different types. Okay, the cross value of different types. Initially cross, you get F1. So you can go for back cross. Okay, so back crossing is what? The F1 into either of the parent, A or B. Okay, then you can go for selfing. So selfing is not possible in human being. Okay, it is unethical. Okay, it's not possible as well as it is for only plants. Okay, so then your haploids, double haploids are those what we usually from F1 we go for, and that also, and then from using processing or some chemicals, you can go for making double haploids. I'm doubling the chromosomes. You generate haploids, then go for. So this is your biomedical process. So association mapping. So here the germplasm or accessions or the breeding lines we take here as an example. So what happens? So we know in say Assam Agricultural University we have 4,000 germplasm. We have about 4,000 varieties. So these 4,000 varieties might have come from different parents. Or through the process of evolutions, they got some distinction. So naturally they have some recombination hotspots. Naturally they have recombination hotspots. And those hotspots can be picked up through a mapping method called associational mapping. We are not going to discuss in detail because it is not within the scope of our lecture. So this is what these germplasm accessions can also be utilized to detect PTL. I mean, when we talk about how to map, we can do that. So there is one of the ways, but we cannot develop a variety. We cannot directly go developing a variety through association map. We cannot go. Or we cannot directly develop, but we can develop a variety if we go in one of those. Okay, try to release. So again, question comes: why we should go for PTL detection here again? Okay, so because our target is what to develop certain variety and to substantiate the farmer's income, okay, or to assist the food, hunger. So in that case, why then we go for association mapping? So these association mapping will give you the first-hand information, first-hand idea that there are some germplasms, there are some, even languages or genotypes that may give or provide certain resistance to certain diseases, maybe stress resistance, abiotic stress resistance, maybe quality wise is more pronounced. So those kind of information you can generate. Okay. Today we are going to discuss about how we can go up to a variety, right? Sorry. Now this is what exactly we go for in linkage versus association mapping. Okay. So this is what, as because we are going to utilize more and more number of individuals from the ancestral, recombination breakpoints. We are going to utilize. So that's why this will be having higher resolution, sharp resolution. Sharp resolution, but here the resolution will be a bit less as because there are only two parents involved. Here, if you compare each one as to be the parent, that is multiple parents are involved. Okay, from different ancestry. So that's why resolution may be higher. But directly, we cannot say this QTL. So this QTL, if you obtain here, we cannot directly say that whether it's from this parent, that parent, other parent. We are not sure. That's why the main problem here it is. So we cannot go and tell that, okay, this, say, blast resistance or say any disease resistance is coming from this either of the parent. We cannot say that. But here we are sure that this is having the trait. So any resistance, it is showing up definitely from this parent. So that is why, it has got a validated one. Here we need to further validate. Okay, so that is, you can say, a first step towards. So once you get it, so there are two ways for complex trait management. One is first you can go for association mapping, next is your linkage mapping. That is your using biopharmaceutical method. So some people have interest in going through this because of this basic needs or scientific needs. Okay, just for your scientific appetite, people try to scientists to go for this kind of experiment. It's good. Okay, but personally, I don't have a very optimistic opinion about this. So I resort on this. Okay, so I have been engaged in mostly is biopharmaceutical marine populations and technical detections and further improvement of the. Okay, so this I can say it will give you a first-hand information. Okay, so good. Okay. So this is the assessment mapping outline. So I'm not going to detail. So if after the class, if you still are interested in, I will just describe. Okay. Now I'll just keep it like this way because again it will be a long story to tell. Okay. So after the class, if time permits, I can discuss some more and more. No problem. Okay. Now coming to the mapping of details using biopharmaceutical mapping population. Okay. So there are some requirements. What are those requirements? So one is your molecular marker. So I told you what is a molecular marker. So the characteristics of the molecular markers to be utilized here is the marker has to be polymorphic and codominant. Two basic things, apart from all other features. Codominant and polymorphic. So I'll come to this point, what is polymorphism and what is codominant marker. I'll just come to this one later on. Okay. Next is your mapping book. Next is your mapping population. Okay. So there are different types of mapping population I showed you, right? So these are different type of mapping populations. Okay. So this is your mapping population. Okay. You'll be required. You'll be requiring extensive genotyping and phenotyping facility. Okay, genotyping. Genotyping is what? When you go for marker analysis, that is your genotyping. Next is your phenotyping. I mean the trait that you are going to suit. Say if it is biotic resistance, say you are going for a pseudomonas resistance. Say your particular or likely resistance. So you have to inoculate the particular variety, and then you need to check the difference among the genotypes. Okay. Now, statistical interpretation, which is more crucial, so we need to go for statistical interpretations. So these are the requirements. So what is molecular markers I described? Mapping population is a crossing population derived for particular trait of our interest. It may be F2, I mean second filial generation. It may be recombinant inbred lines, back cross inbred lines, or double haploids. Okay. So what exactly we do? So we go for genotyping and phenotyping of the mapping population. Okay, any of the mapping population F2, it may be recombinant inbred lines, it may be double haploids, it may be back cross inbred lines. And we will go for genotyping and phenotyping of the mapping population together with parents. This is very important. Parent has to include. Without parent, no analysis. So parent will not happy with it. Okay? Parent has to be there. Now, then we need to find out the recombination frequencies. Then we try to find out the significant correlation between the maximum recombination point and the phenotype. And then we go for, after utilizing some statistics, we will try to establish that there are tightly linked. So there are certain statistical parameters based on which we say that there is a clue. Okay. So one of the parameters, so I am not going to give in detail. So one of the parameters is load. Load. Early morning, this is load. Okay. So load score is what? The version of what? Logarithm of odds. Okay. So what is the logarithm of odds? What is logarithm of odds? So LOD score, LOD, a capital LOD. Logarithm of odds or logarithm of all ratio, because it is a ratio. Ratio of what? Log 10 of probability of getting a QTL at a particular marker interval over probability of not getting a QTL at that marker interval. Okay? Log 10 or probability of having a QTL at particular marker interval, say X, over probability of not getting a QTL at that marker interval X. Okay. Now, usually we say, so each of the marker interval, because we will be utilizing, say, hundreds of markers. So our hundreds of markers, we will be having many nodes. So from there, we need to find out the threshold. Threshold node score, and then we can declare anything more than the threshold node score will be your. So that is, usually we say that is the QTL. Now question comes, why? It's a probability based thing, so whether it will be that kind. So to substantiate our probability level, we again go for another parameter called regression. How affinity, how tightly they are. So I'm not going to discuss because it is all statistics part. Okay. So this is your mapping population, right, in a pictorial way. Okay. So double haploids, how do you perform? This is your F2. Okay. This is your double haploid, this is your recombinant unit lines. Okay, this is your near isomeric lines. Okay. These are the mapping population. Now, I say molecular markers, so those molecular markers have to be what? Polymorphic. Okay? So it has to be polymorphic. So this is an example that it is not polymorphic because it's showing the same size of the band. This is showing, say, parent 1 and parent 2. Say if the development of varieties Ronjit someone. So Ronjit is one variety, another is just one someone. We are making a cross. So this marker is not useful. But this marker is useful because these two parents are being clearly visible. Okay. Next, I say codominant marker. So this codominant marker is a type of marker that can differentiate capital A, heterozygous, and this homozygous recessive individuals. All these should be differentiated. If you use dominant marker, either of these can be differentiated. That means what? Exact ratio we are not going to spot that. Okay? The exact ratio we are not going to spot and we are getting erroneous results. So because of that, these two criteria has to be fulfilled, apart from all other criteria. Okay? So this is what the requirement. So once we do the codominant marker, so we can utilize various markers, and there are different individuals. Okay, so one is A type, one is B type, and this H represents what? Intermediate or heterozygous. Okay, so likewise we will score these. These are different, say, individuals, and these are different markers. Okay, fine, and we are making an Excel spreadsheet. Okay, then we will go for find out this linkage map, chromosome wise linkage map. Each of the markers are being, or each of the positions are being associated with molecular markers which are codominant, polymorphic for the parents. Okay. Then what exactly we do? We construct the mapping population, P1 and P2. We go for genotypic data and we go for phenotypic data, that is your quantitative data. Okay, say trait, the level of tolerance. We need to evaluate. Okay, we evaluate the level of tolerance. Then go for statistical analysis and find out this kind of ANOVA regression or permutation, like your low score. We find it out, and then we go for significant association. And when there is significant association, we can declare that there is a QTL. See, all the marker intervals are not showing a good peak. This is your threshold. This, horizontal bar is showing the threshold. So this threshold value, anything above this 2.7, we are considering that there should be a QTL. So there is a QTL in between these two marker loci. Okay? These two marker loci. We are having the QTL. Okay? So that's how we detect the QTL. Now question comes, is it all that? That we are having QTL and it's all fine? Then now we have wind up our job. Should we wind up our job or should we continue? So what beyond this QTL detection? QTL to follow, that's all fine. We got a QTL. Now what next? So once you get a QTL, we can go for multi-stage selection or cloning of the QTL. We need to go for cloning of the QTL. Okay. We can clone or we can go for multi-stage selection. So what is multi-stage selection? It's simply just to assist the normal breeding process. Sorry. So multi-stage selection refers to the use of DNA markers that are tightly linked to the target loci as a substitute for autostage phenotypic screen. So to apply multi-stage selection, we must have a QTL of measure effect. Unless we have a QTL detected, we cannot go for multi-stage selection. Multi-stage selection we have to have a QTL. Then we can go for. And what exactly does in a conventional plant breeding? Okay. So this is your parent and this is your P2 and you have your F1, F2 and large population. Okay. Then you go for your phenotypic selections, your glasshouse trials or your field trials. Okay. So all those you will go through. It's a huge cumbersome process. Huge cumbersome process. And if you want to minimize where this multi-stage selection can play a role. Another point. So now, what multi-stage breeding can help you in this regard? So what happens in multi-stage breeding? Say this is your parent, okay? P1 and a P2. P2 is having say 200 base pair band. It is showing 100 base pair band, lower one, right? So resistance is showing 100 base pair band, for example, it is showing 200 base pair band. Now, this is your F1. Now, you might be having confusion in that. So, why there are two? F1 has to be intermediate. Does anyone here in this house have the opinion that F1 should be in between 200 and 100? Does anyone? Does anyone have an opinion that this is a 200 base pair, upper one and lower one, so F1 should be intermediate? So why it is giving two bands? Why I am showing here two bands? It has to be intermediate. The F1 usually phenotypically is intermediate, say most of the cases. So do you have any different opinion that it has to be intermediate? No? Then why? Why do they call it intermediate overlays? If I say that it has to be intermediate. You all agree that it has to be two, or you have some different opinion. If you have, if you have an opinion that it has to have two bands, then why it has to show two bands?
It is genetically heterogeneous.
Genetically heterozygous. How can you say this is genetically heterozygous?
Suppose for capital is smaller, it can have the both of the capital is small in that one. That's why there are two bands.
No. What will happen in the middle one? So if I say like there is no band and only the middle one, 200 and 100, and it has to be 200 plus 100 divided by 2, it has to be 150.
Maybe in the that time those bands are linked, so because they are linked, they segregate separately in their part. So they didn't cross them. That means that they are linked.
They are linked. That is right. They are linked. Okay. So she said, because there are two different, and that's why we are getting these two different. But usually mathematically, if I say 200 plus 100, okay, divided by 2, which is 150. But we are not getting it. Exactly, you don't get it. It is not mathematics. It is biology. Why? Because it is defining a parent. It is also defining the other parent. If it is an intermediate, whether I be sure that from where it is coming, which allele is going where, it is very much required, even your parental identification. Okay, say if I am to be sure that I am from my product of my parents, if it comes in between, so the result will be completely misleading because it will be neither ka nor udhar. What? So that's why you have to be very careful. The days are coming when you may be required this kind of, especially for forensics. So that's why forensics in a crime scene. So this helps. This is the example of co-dominant marker. This is a co-dominant marker. Dominant marker cannot show up. So it is not 200 plus 100 divided by 2. It is not mathematics. That means what? If it is susceptible, say SS. One is coming here, one gamete coming here, RR resistant, R is coming in. So small RR or capital RR, so R, this is your capital R, this is your small. So you have to know that exactly that the allele is coming. So that's why it has to be there. So you might have run a PCR, so if you have two individuals, two different fragments, you mix them and loading them there, across there you will get this kind of results. Okay, so that is what. Now coming back to this. So what it will give us? Okay, now the selection part. You see, I need to grow the plant, but before growing the plant, what I do from the seed, I isolate the DNA and use the marker and go for genotype. What about QTL is linked? I know the QTL to select. I know the marker positions where it is showing up. So I will go for, raising the plants in a net house, in a greenhouse, or in field. Before raising in the field, what I'm going to do, I'm going to find out which one is exactly needed to plant. Okay. Now tell me, which plant I should plant? Which should be more important? Which plant I should select? Tell me. This one? Yes. This one?
Yes.
Yes. This one? This one?
Yes.
This one?
No.
No? Okay. This one?
Yes.
Yes. Yes. No? Yes. All of you are saying like this? If someone asks me, I don't want to waste, many years. Just give me a variety, I will just choose this one. This one. I will choose this one. Okay, sorry, no. This one, only the one showing up. Okay. Because it's already been crossed. This is no more P2. It has changed its genetic morphogenesis. It's already, gone. Identity has lost. That's why people say when you get married, everything is lost. So that is why. So is what, I mean, it is nowhere else. I mean, it's not, I mean, you are getting this band, but it is representing what? It's F1 only. We only confirm that it's F1, 2F1. So this is the segregating F2. Okay, now only I need to choose that. But if I want to have more and more, definitely again I will choose this and plan it for another generation. It will segregate again 1:2:1. Okay, then we can go for. That is the beauty of utilizing this technique. So your job is, your job is what? Almost 50% reduced. Or you can say if you go for 1:1, only out of 4 you need to plant only 1. If there are 4,000 plants, only 1,000. So your jobs are getting reduced. Okay. So that is what a multi-assay selection is. Okay. Now I'll take only 3 or 4 minutes just to take you to the real arena of what exactly going on. So you can find PCL papers more than every year more than 3,000 papers. More than 3,000 papers a year. Here across board. But hardly we get 1 or 2 varieties. Especially on contracts. Okay. So whether all the working is in vain or we are going through a process that is finding a needle in a haystack. So what else we are going through? So actually it looks very simple. As I said, the train itself is complex. Train itself is complex, then how can it be that much simple? Okay. So there are a lot of obstacles. Okay. The major obstacles I want to talk about is what fetal cloning is a very tough nut to crack. Very tough nut to crack. Why it is? Because of this. Okay. Because of which the Titanic has lost its way. Right? Because our target is somewhere what? Here. But we don't know there are a lot of hidden effects. There are a lot of things pulling and pushing each other. We call it as epistatic interactions. We call it as some other interactions. I mean, there are push-pull effects. No one is allowed. Like our society, if someone performs well, they try to drag him or her down, so that the other can shine, right? Like the Hima does. Okay, when she was performing, everyone is applauding. So when she was not performing, everyone wants to press her down. Okay, likewise. So here, so target is somewhere here, but there are a lot of hidden effects. That is, very difficult to manage. So if you can manage, yes, you are going to get it done. So the reasons, as I say, resources, equipment you need to need, markers sometimes not cost effective, accuracy of the QTL, mapping studies, sometimes QTL effects may depend on different genetic backgrounds. Some genetic background it may work fine, some other genetic background it may not work. Of that, I'll make capacity. So Ranjit Saban, Ranjit background it was working fine, in Bahadur background it was not working to that extent. So likewise, when the background is changed, when the parents use change the parent, the effects may get down. Okay. Sometimes with lack of marker polymorphism or poor integration of molecular genetics with the conventional breeding or conventional breeders. There are some reasons that I wanted to jot it down. Okay. So this is what the main one is what the lot of minor effects with the epigenetic interactions. Because of all of the interactions, we are not going to get reach that unity. Okay, that is the main point. So this is your take-home message. We know the traits are complex, tough, but not impossible to manipulate. Okay. So what we need, we only need technologies to improve. So things are improving. So for last 20 years, when we did my master's in 2002, so the technology that we were working with, it's all become almost obsolete. Newer and newer technologies are coming up, nobody knows it. Okay. Once people thought that protein is a genetic material. Now we know that DNA is a genetic material. So things are changing, things are moving in the right direction. We are hopeful that we can get more and more varieties like the soda sabon or Ranjan sabon. Okay, this is for basically for submerged instruments or plant resistant varieties. So there are lots of varieties because people have understood that the evil plateau you cannot break. But situation specific, quality wise, situation specific variety needs to be developed. We cannot go in 1960. We get a jump from 2 tons to 5 tons variety, from 5 tons to 7 tons at least for hybrid rice, but 7 tons for last 20, 30 years. It's not that easy to break the evil plateau, but situation wise, it's a drought resistant. This area is on the drought prone area. So that we can introduce drought prone variety. Flood prone area, we can introduce flood prone variety. Blast, in the prone area, we can introduce blast prone variety. Likewise, we can plan for those specific situation specific varieties. So this is what the technology, when it meets the reality, or this is the best example that I really ever found. This is the same farmer, Subara. Okay, he is cultivating Swana Sabwan, and this is the Green Revolution rice IRA he is cultivating. Okay, so what technology makes, or you can see this, really, science makes stories. Okay, so this is the beauty of science. So this IR8 is the first high-yielding varieties of rice. Then the same Subara is cultivating, Swana Sabwan. So technology, when is adopted by the end people, then the real beauty of science or any technology flourishes. So that is what I wanted to point out. So that is what technology is there that we need to utilize judiciously. We need to implement it. Government has to really need to move forward. Okay, to allow the scientists to adopt the technologies. There are some technologies like GM foods and all. They are not governments are not all governments are reluctant not to adopt that because of different legal and other social litigations. But yes, the years to come, we need to go forward because at the rate that population is rising, we need to adopt certain this kind of efforts have to be made and in the direction, and we need to be really accustomed with those kind of new and newer technologies so that we can better benefit the farm. So with that words, I'd like to conclude. Thank you for your patience. So any questions? Any questions? Welcome. Or even after the class, we'll be having class, so you can jot down, you can write my email. You can ask if you find any problem regarding this. It's a 1 hour class, so we cannot finish everything. Just want to have a glimpse of that. So still if you have some queries, I know you're all tired. You went there, you came back, you had a class, and then you went for practical again. Having a lecture at 4 o'clock is not that important. It's not happening actually. At 4 o'clock, everyone wants to go back home. 4 o'clock, having this kind of lecture is again a worry, but still if you have some questions, otherwise, you feel free. You can go home without any problem with any permission from your coordinator. So I want that from my side. So you have any queries?
Anything?
Not anything. What are you
I want to ask you a question, but that is not related to this subject. You got an next scholarship, you want to apply for them. Okay, so how was your experience? How did you get there? What is the eligibility criteria and the requirements?
Okay. Max is really advertising. You all can go for all the government scholarships that have been advertised in education.nic.in. There's a portal, education.nic.in, or in Ministry of Higher Education. There is a special, tag for scholarship. Scroll down the notice, you get this one, scholarship of fellowship. Okay, year round it is going through. So Minister of Higher Education, there is an advertising employment needs as well. Okay, all the government scholarship. Okay. So before that, it's a year round actually. Chinese government scholarship, then first it comes to Chinese government scholarship, then math, then Commonwealth, then it comes your Irish fellowship, then there's a lot of like Commonwealth, Canada will come up. So these scholarships, so basically for math, what exactly required, the time they publish, so you need to have approval from the, one professor from there, and then if you have, approval is better always, but I didn't have that approval. I didn't know that because in 2002, internet was horrible here, okay, and we had to go for, CSC, those centers. But the procedure is, there is a university selection, internet selection here in India. Then they will nominate the names the Japan Embassy. Japan Embassy will contact the respective organizations or the teacher, and then the teacher is he or she saying yes, the communication will come to the Japan Embassy from the Minister of HR. Okay, so nowadays Minister of HRD has opened up that the examination part for him. Like very simple English examinations and some very basic questions. Nowadays, even last week, there's one of my students applied and she got qualified, she got a job and then she stopped. But there is a first group, okay, through embassy. And HRD, embassy, then it's an auto process. There is a one program, if you contact directly the professor in charge. Like say, Japan Institute or Horse Institute, if you write for it, this simple one. Okay? You can contact the professor directly if he is willing. The main condition is that if a Japanese host professor is having Japanese students, it is reluctant not to have the foreign students. Because they are finding that. They are more comfortable with that. Usually, those teachers, you can search for those teachers which are and who are not having enough Japanese students in their life. They can write to the Max in Japan and they will directly call you for interview and pick you. Okay, so that is the best option if you can have it, but you are just bound to your professor role. These activities may be very clear, but if you go from here, you are the boss. Mostly, you are the boss. Okay. Otherwise, there are two ways. One is from this, another one on the other side. Okay, and always try for national university. So the professor from national university, he writes, then your this one is true. So say I went to Hokkaido University, I didn't know because my project proposal is almost the same that resembles with the one that I submitted. And it resembles with that professor. Okay? And later when I went there, then I came to know that he was a DBT, he is a bishop. I didn't know. You can write like, I didn't have any communication before I went there. Okay? Because that time internet was not that. That's because of. So then, even in science fellowship, they didn't give more, fellowship is almost the one that you can survive. Very expensive, but you can survive. Science fellowship, you can write for Commonwealth fellowship. Even European educations are not expensive as your budget. You might be knowing, right? You will know that. But you always try to have a better education or basic education here in a country where your degree is certified. Otherwise, it will be problem. Most of the cases, most of the countries, European countries also, they don't have master's degree. From directly from PhD, they have a 1 year MSc, which is not being recognized here. So do your MSc, your postgraduate here, then you will plan for. And it's cheaper. You go there, 1 year it may be difficult, but then you can go for, because a lot of our friends, I mean, in that time, someone went to Australia, someone went to first year was struggle. If you work hard, definitely you will get some backup scholarship. Okay. But I don't suggest that, unless you get a government fellowship, you should not get the money. It is very expensive. So without having a permanent solution about money, you should not go. But Europe, you can go for part-time jobs. But here, any part-time job, because of the language problem, it will be really hard to learn without money. So that's apart from this.
Why we are not yet a minor effect 2 years?
Minor effect?
Yes, a minor effect of the.
Oh no, the minor effect you can later. We have to see minor effect because if you want to,
They are also running.
Proceed. All matter is here. I don't care about anything, what benefit is going behind. I don't care. I want doctorate, if I get doctorate, that is fine for me. If I get doctorate with a method effect, then that is fine for me. Because I am not handle all those. So if I am getting doctorate with maintaining those minor effects, then I go for minor effect. Otherwise, I don't. For my case, trade is important. What about? Yes, minor effect may contribute. But if it is not significant, why I should do it? Why I should do it? Minor effects, we should not eliminate. If it is some way or the other way is there, or contribute. Say, I am getting 10 kg of rice, applying this technology, or manipulating say 1 locus, I am getting 10 kg. And if I am getting 11 kg by manipulating 12 meters, which are I choose? I will replace with a simple one, 10 kg. It's something will be 11 kg. 1 kg is different. And I have to consider 11 meters. Why I should consider? I have to consider. That is important. One see, you just believe. Okay. So because even if you handle this, complexity or the behavior of the trade is so complex that it may change. They say that across the environment, the yield is almost a part. I consider that one. I don't consider about those. Even if you say that this video is showing this low score, this showing this mark effect, I don't do it. And I think others even bidders should know that. Because bidders are not good. That's why there is a so science is some other part. Science is there. Science has to be there, but science is related to what there is. Okay. So minor effect definitely should consider, but unless it doesn't show or does not make any significant difference, I don't bother. Personally, I don't bother. Technically or theoretically, I should bother. But I should. If you talk about what did I say? Trade value point of view, it's better not. কেনেকুৱা লাগিছে ঠিকে লাগিছে বঢ়িয়া চলি আছে খানা চানা ঠিকে আছে
বেছি কৰে
বেছি খাইছে
খানা বেছি বেছি
Thank you so much. Anyway, thank you so much.
Thank you, Akhil. Thank you
No
so
problem,
much
no problem.
for your nice presentation. I was listening online there, and I know he's a very good scientist, so he knows everything very nice.
ওর উপর সাইড দিও ঠিক আছে
और ऊपर नीचे उसी को से क्या लॉजिक है ठीक है Thank you. Tomorrow morning we have a lecture by Mr. Akhil. So, it was my general homework. He is going